Thank you for the introduction, and thank you for the opportunity to share our research here at MPS this year. My name is Raquel Ajalik. I'm a graduate student at the University of Rochester working under Dr. Hani Awad and Dr. James McGrath. We'll be presenting our work towards developing the human tendon-on-a-chip platform that is a model that is compartmentally designed to look at vascular and tissue interfaces that model fibrovascular injury and therefore healing. So fibrosis is defined as a thickening and scarring of connective tissue, and it usually results either from injury or the ongoing diseases. And when it affects major organs such as the heart, liver, or even the skin, it can really be quite detrimental and can be a large progressor of disease and ultimate mortality in many patients. And unfortunately, there are zero current treatments that either inhibit or reverse the effects of fibrosis. And so given the expertise in our lab group, we used the tendon acute injury, sorry for the graphic picture, acute tendon injury to follow this fibrotic disease. And acute injury to the hand flexor tendon, or in tendons generally, really affect very young populations, and it's characterized again by a scarring of the tissue. And so we see in the histology image on the bottom, there's quite a dysregulation of extracellular matrix, very high proliferation of cells in this environment, and what this leads in patients is a very decreased range of motion, very high rates of re-rupture, recurring inflammatory flares, and ultimately very high monetary costs for the patient for the rest of their lives, and therefore a large burden of this disease overall. And so what we have done is to try to study this this pathogenesis, and in this histology image what you can appreciate is that pretty much like in any healing, we have a large infiltration of blood vasculature. This vasculature enters the injured tissue, and there is of course a influx of immune cells. These immune cells are very important in propagating the different healing stages, and really their communication drives a lot of different responses. In fibrosis in specific, it modulates the differentiation of the resident fibroblast stromal cell and differentiates it into an activated proliferating myofibroblasts. And these myofibroblasts are the active disease state. They are really doing a lot of the organization in the matrix. They are progressing the disease and ultimately lead to these endpoints for these patients. Now, chronic inflammation really pushes these myofibroblasts into a chronic state known as the senescent fibroblasts. These senescent cells aren't really replicating, but they are large signaling cells, and they secrete senescence-associated secretory proteins, and this milieu is really what propagates and really attenuates the resolution phase in the tissue. And so this is the interface that we study, and even though there's a lot already known, there is a lot that's still needed to be studied, particularly the differences in contributions of resident versus infiltrating cells and what their paracrine signaling is important in the progression of this disease. Particularly in tendon research, a lot of this work has been done in murine models, and it's actually great because we have a really large database that we can compare our results to, but we really want to isolate and tease out what our data from murine mouse models can really be used in human translated research. And so we're really working side by side, particularly in our lab, with our murine model, our in vitro model, and ultimately human translation of what we learn. And so our main objective, again, is to develop a human in vitro model to really study this pathogenesis of fibrovascular tendon healing with an ultimate translation objective of using it as a therapeutic screening platform. And so, as I mentioned, the interface that we are modeling is disease. And so we look at an inflamed endothelium interfacing with a fibrotic tissue. And this is really simulating a very small cross-section of the infiltrating vasculature with the surrounding fibrotic adhesions that you can see in the green in the histology here. And so the device and platform that we use, we build in-house. It is a device that's completely made out of acrylics. We really want to decrease the absorption rate of anything that we put inside. It's kind of like a transwell, so it's mimicking that well interface format. And it is very small, so we we've designed it to be arrayed into a 96 well format, and it's currently being outsourced, manufactured through A-Line and Simpor, so we get very high reproducibility in our devices. So the top component is where we house our porous membrane, and that's how we communicate between the top and bottom components. This porous membrane is a dual porosity. So we have 5-micron pores etched into a silicon nitride membrane, and these 5-micron pores are ideal for cellular extravasation of anything between the components. These 5-micron pores are patterned on a nanoporous environment or surface. And these nanopores are really great because they're very small, they're under 60 nanometers, and they are perfect for paracrine signaling, and they allow for protein diffusion between the two compartments. And this substrate is amenable to cell culture, so this is where we grow our endothelial cells up to confluency. In the bottom channel is where we house our tendon tissue construct. This is a collagen hydrogel where we seed any cells that we want to study on the tissue side. Again, we're mimicking tendon architecture, and so we really wanted to incorporate native anchoring that is known in tendon as it connects muscle to bone. This is very important for two particular purposes. It helps, of course, to mimic that architecture, but the cells are very, very responsive to extracellular matrix cues, and so this directed strain that is allowed upon the cells restructuring the matrix and being anchored to these 2 points really helps in their differentiation in that myofibroblast phenotype, as you can see here through alpha smooth muscle actin staining. And so we combine these top and bottom components reversibly. We connect the endothelium to our tissue construct, and this is done just through PSA adhesion between the two components. This is great because it allows for reversible binding. So at any point in the experiment, if you only want to study a single section of the device, you can take it apart, play with it, put it back together, whatever it is that you need to do. And so I'll walk you through a little bit of what our workflow looks like in developing these platforms. The modularity really comes in handy because we want to look at the hydrogel and build a model that is really, looks like what we see in vivo. And so the main cell types that we look at are tendon-derived fibroblasts, which are called tenocytes. We embed those with monocytes that we obtain from peripheral blood. But we obviously, monocytes don't exist in tissue, so we want to polarize them into a naive phenotype. And so we co-culture these two cell types in the presence of monocyte colony stimulating factor for 6 days. And then on the 6th day, we also introduce endothelial cells onto the top component. This step is done independently because we want the cells to reach confluency before touching the tissue construct. That after those 24 hours, we attach the top and bottom components, and this is where the magic starts to happen. So we introduce the monocytes, freshly isolated monocytes on top of the endothelial cells, and that is mimicking the arrival of immune cells to the site of injury. And to induce a fibrotic injury into the tissue side, we introduce transforming growth factor beta 1, which is a large mediator of fibrosis in vivo. And so this is, at this point you have the complete device. We culture this for an additional 3 days, and we take different endpoint analysis. This whole setup is performed in serum-free conditions, so we are really trying to control as much as we can at this point. And so again, this is a model of disease. So on the vascular side, we have an inflamed endothelium that we've characterized by upregulation of leukocyte adhesion markers. We also see dysregulation in the injury site that is prominent of inflammation and angiogenic responses. On the tissue side, we've performed analysis to see what these populations look like in these cells. Again, we don't add any exogenous cytokines to really polarize these macrophages into any type of phenotype. So this differentiation that you're seeing into pro and anti-inflammatory macrophage types is really just due to cell-cell crosstalk and their communication between them. Excitingly, we see upregulation of important markers such as TD206 on these macrophages. This is a important marker for tissue-resident macrophages in specific. It's really involved in the apoptotic response in a lot of these cells. We also looked at the cellular construct population. We add these cells, the monocytes, with the fibroblasts at a 1:7 ratio. This is mimicking 1 week of injury in the tendon tissue. We got that from literature results, and we can see that even after 10 days in culture, this is still maintained. So we are can maintain these two populations pretty well. Again, that SAS profile that I was talking about, we are probing for that, as we can use that to get a sense of how the culture is responding to its environment and how the disease is progressing. So we simply show 4 of the many cytokines that we tested here, but you can appreciate that by 24 hours we have a pretty developed SAS profile, and then these are maintained at the 72-hour time point. This is very different compared when you just culture tenocytes alone in a hydrogel, which is very commonly done. We performed additional analysis to see who is actually driving this secretory profile, and we identified that that monocyte that we add onto the vasculature side is really the main driver of either inducing the secretory profile in the other cells or itself secreting a lot of these secretory cytokines. And again, it plays a very big role because it's activating the endothelium itself. It could be transmigrating to the bottom construct, so it's very, very important player. And so these metrics so far I've showed is just inflammation and fibrosis metrics that we tested ourselves, but of course this is a disease model, so we need to compare it to human data. And so we used data from bulk RNA sequencing out of the Rouen group that compared human tendon tissue as well as human tenolysis tissue, where we were able to perform our own bulk RNA sequencing in our HTOC platform and compare that to the Rouen group's data. And here we wanted to assess comparisons to see how close we can get essentially in our simplified model compared to real in vivo disease. Turns out we can do pretty good. We have a really high correlation between extracellular matrix pathways and regulating those. Very excitingly, we have a lot of overlap in interleukin signaling. That is, again, not surprising because a lot of this is what is propagating ultimately this disease. And we do have some gaps in places for improvement. So some of the immunoregulatory pathways related to cells, such as T cells, natural killer cells, and dendritic cells, are opportunities, I think, that we could be adding into next generations of the HTOC to get a fuller profile of this disease. One of the pathways that RNA-seq elucidated into being targetable for the attenuation of fibrosis is the PI3K-AKT-mTOR pathway. And there's a lot of murine data actually correlating this pathway with tendon fibrosis. And so we tested to see if it would be upregulated and possibly targeted in our model. And so we looked at particularly downstream 4-ABP1 and S6 effector molecules of this pathway, and we noticed that we did get upregulation of these in our quad culture model. This is very exciting because now we have a way to go and a direction to go into. If you are able to attenuate this mTOR pathway, you can control how these fibroblasts differentiate into myofibroblasts and some of that protein synthesis that is going on in the environment. And ultimately, we can find a way to improve resolution. This is not a problem simply to tendon, and so of course there are already therapeutics available for such pathways. Importantly, rapamycin is an inhibitor of the mTOR pathway. It's already FDA approved and has been used in different immunoregulatory cases, even in cancer. And so we tested this platform in our model to see if we could monitor some of its effects. The way we do this is we introduce the drug onto the vascular side. This is mimicking intravenous or oral arrival of the therapeutic. We only cultured it for 24 hours in this initial analysis, and then we do very similar endpoint assays. So I'll just be presenting a few of our data on this. We first look at our vascular side, and what we notice is we have drastic downregulation of that S6, which is one of the 2 downstream mTOR effector proteins. We have slight attenuation of 4-ABP1, but not completely. But importantly, we started seeing some improvements of the vascular monolayer and its integrity and cohesiveness. This was verified by junctional staining of VEEC adhering, and we noticed that some of the gaps that were introduced initially in our injury model were somewhat resolved upon the rapamycin treatment. And this is very important because rapamycin has never really been tested on its effects on vasculature reaching the injury site, so we always look at the endpoint of if the tissue was resolved, but this could be a very important aspect of the mechanism of action of rapamycin in vivo. On the tissue side, of course, we started looking at that too, and we saw that at 24 hours we do see some attenuation of myofibroblast expression, meaning that some of that rapamycin that worked its way down to the tissue side was able to do a little bit of work on these cells. We looked at the transcriptome of this treated platform, and what we noticed is that the genetic markers for mTOR as well as some of the inflammation markers were starting to come down. But what we didn't see is the change in the extracellular matrix genes that are responsible for a lot of that organization. And what I think this is happening is because our time frame is so small of just 24 hours, usually you see rapamycin treatments in very long-term administration, and so we are looking into multi-dosing and introducing rapamycin over longer time periods to see if we can get some of that fibrotic phenotype changes. So in summary, we've developed a platform that captures paracrine signaling, immune cell infiltration, and drug response in the context of fibrovascular injury. We were, I think, provided a method to validate these in vivo disease in our in vitro model simply by comparing it to published results. And ultimately, our context that we're looking at is this vascular tissue interface. And this injury looks a lot like injury in a lot of organs and a lot of tissues. So if you can simply move or replace the cells that we are looking at specific to tendon, you can really model this disease in many organs that suffer from such fibrovascular injury, and ultimately be able to find efficacious treatments for fibrosis. So with that, I'd like to thank my lab groups. This collaboration effort is 3 labs in 1, and I just presented a very small snippet of what is going on. There are a lot of abstracts here that are looking at in the introduction of fluid flow, how we can sense these SASPs over extended time periods, as well as machine learning integration of like tracking the immune cells, and how to really improve the matrix. These cells are very, very responsive to extracellular matrix cues, and so we really want to improve the fidelity of that. Additionally, I am looking for job opportunities, so if you are interested, please hire me. Thank you.
Thank you, Raquel. I think you're soon hired. Are there any questions?
Thank you, very nice work. I had a question simply to know if you ever checked on the effect of mechanical stimuli on the inflammation, fibrosis, and also because you just said that ECM is so important. Of course, that has an effect on it as well.
Yeah, that is another avenue that we're currently incorporating actuation on the tissue side. Actuation is very important and actually a big question in the field of when to introduce movement to improve healing. And so we definitely are working on incorporating actuation on our tissue side.
Yeah, hi, from Pittsburgh. Good to see you again. Great work as always. In your study, comparing data to humans, is it a single arm sequencing or bulk arm sequencing? Because different cell type in your model, right?
Yeah, we did bulk RNA sequencing. It's a really interesting point that you bring that. So the Ron group did isolated the tissue, the tendon tissue, and then performed bulk RNA-seq. In our model, we performed bulk RNA-seq on the vascular compartment independently from the tissue compartment, and we had really, really cool results of that. But when we tried to compare that to the Ron group, we needed to pull everything together to make it a better representation of that. But we can talk offline to see some of those results. Yeah, thank you.
Short question. Yeah.
Working down the line here.
Thanks for the presentation. Is there relevance to integrate the vasculature within the tendon tissue, or have you looked into it?
Yeah, that is the following direction, of course, to see the angiogenic response to see angiogenesis. So yeah, we're very curious into seeing how that responds. It's a different model than what we're looking at here. This here is a simple cross-section, and I think there's a lot to learn at this point here. Introducing infiltrating vasculature into the tendon tissue, I think, introduces variables that are hard to control in vitro models in general. And so it's definitely a stepping stone, but I think there's a lot to study and to learn just in this model so far.
All right, thanks. Next, yeah.